Telomeres, the protective end caps of chromosomes, have emerged as a central focus in the science of aging and disease prevention. These repeating DNA sequences, though non-coding, play a vital role in maintaining genomic stability. Each time a cell divides, telomeres shorten slightly, a natural process tied closely to the aging of cells, tissues, and ultimately, the organism. Once telomeres become critically short, cells either enter senescence, a non-dividing state, or undergo programmed cell death. This progressive shortening serves as a biological clock that reflects not only age, but also cumulative stress, inflammation, and lifestyle factors. The correlation between short telomeres and many chronic diseases is striking. Cardiovascular disease, type 2 diabetes, neurodegenerative disorders like Alzheimer's, and even certain cancers are more common in individuals with shorter telomeres. Moreover, studies have shown that people with unusually short telomeres for their age are at greater risk of early mortality. These findings have led researchers and biotech companies to explore whether slowing or reversing telomere shortening could be the key to extending health span, the number of years one lives free of serious disease. Encouragingly, recent research is uncovering ways to influence telomere dynamics. One approach centers on activating or enhancing the activity of telomerase, an enzyme capable of rebuilding telomeres. Telomerase is typically dormant in most adult somatic cells, but it's active in germ cells and certain stem cells, contributing to their longer functional lifespans. By reactivating telomerase in targeted ways, scientists hope to repair the telomere damage that accumulates with age without triggering the uncontrolled cell growth seen in cancers. Experimental therapies using telomerase activation have already shown promise in mice, with some studies reporting improved organ function, increased bone density, and better metabolic health, without a higher cancer risk when tightly regulated. In human cell cultures, telomerase-boosting strategies have extended the replicative lifespan of cells and restored youthful gene expression patterns. While we are still early in the translation of these techniques to humans, the results suggest the tantalizing possibility that telomere therapy could not only delay aging but potentially reverse some aspects of biological decline. Beyond pharmaceuticals, lifestyle plays a significant role in telomere preservation. Chronic stress, poor sleep, smoking, and processed diets have all been associated with accelerated telomere shortening. On the other hand, studies show that regular physical activity, a plant-rich diet, mindfulness practices, and strong social support may help maintain telomere length. Remarkably, a few small clinical studies have even documented measurable telomere lengthening in people who adopted comprehensive lifestyle changes. This suggests that while telomere biology is influenced by genetics, it is also responsive to the environment, offering a powerful incentive for early and sustained healthy behaviors. Epigenetic interventions are also gaining attention. Because telomere maintenance is regulated by a network of genes, proteins, and cellular pathways, some researchers are investigating how changes in gene expression, rather than gene editing, could support telomere health. This includes compounds that activate sirtuins, AMPK, or other cellular stress response regulators, which indirectly protect telomeres by reducing inflammation and oxidative damage. Telomeres, then, are more than just a biomarker of aging. They are a nexus point where genetics, lifestyle, and emerging medicine converge. Preserving their length is not about chasing immortality, but about extending the years of life that are active, independent, and fulfilling. A future in which age-related diseases can be delayed or prevented through personalized telomere care is becoming more conceivable with each scientific advance. As interest in longevity grows, so does the realization that aging is not an unchangeable fate, but a process we can influence. Telomeres represent one of the most promising frontiers in this effort, a measurable, modifiable target that reflects the integrity of our cells and the choices we make across a lifetime. Rather than treating diseases once they appear, we may soon intervene earlier, preserving health from the inside out by protecting the very ends of our DNA.